Okay, so next presentation is Bennett, also from Baylor University. You can start if you're ready, if the judges are also ready to judge.
All righty, can everyone see my screen? All right, hello everybody. My name is Bennett Shackmuth, and just like Nazil, I'm also an undergraduate here at Baylor University, working in the lab of Dr. Bruce. And my work specifically is on identifying a standard pro-inflammatory activator for human microglial clone 3 cells, much like what he talked about earlier in his presentation. Meant to go to the next slide, hit mute by accident. To get started, we're going to talk about, if it'll let me move over, there we go, the background on microglial activation. So microglial cells are specialized macrophages that can undergo either the M1 or M2 activation upon exposure to differential stimuli. And so these cells are originally derived in development. When your blood-brain barrier begins to form, they are part of the original macrophages. And when that blood-brain barrier is formed, it kind of isolates them from the rest of your immune system. So it's mainly part of the immune system of your brain. And so because of that, they have to be very powerful, but also very careful. So as you can see here, there's 3 different phases they can go into. The M0 phase, which is their surveillance phase, where they're just going around the brain, they're making sure that everything is clear. They can go into their M1-like phase, which can result in demyelination, leakage of the blood vessels, kind of causing like a leaky blood-brain barrier, or the M2 phase, which is part of remyelination and healing. And so that will release anti-inflammatory cytokines, while the M1 phase will release pro-inflammatory cytokines. So the cells that we use for our model are the HMC3 cells, or the human microglial clone 3 cells. And here's another photo we took as well in lab of them when they're at a very high confluence. And we originally purchased these from ATCC, where they have been certified to be primary human microglial clone 3 cells. And they were immortalized via SV40 dependent immortalization of embryonic microglial stem cell or microglial cells. And they're adherent cells, and they're very well used in like the study of neuroinflammation. And that's due to their low maintenance and very high growth. It allows for a very relative ease of stockpiling for assaying. So they're wonderful model systems for microglial cells that are immortalized. So one of the main problems that can occur though from HMC3 cells is their history. They were initially isolated in Paris in 1995. However, since then, they've gone under the name of either HMC3 cells or KIME3 cells or CHME3 cells. And so as you can see here, they've been sent to other labs in the UK, in Sweden, in India, and all around the world, yet they were given different names and they've kind of been passed around. So eventually they ended up in the hands of the ATCC, who certified them and now are the ones that regulate their usage and whether or not they're proper HMC3 cells. So they've been certified to be human microglial cells. And so another part of the problem is that many of the researchers who study neuroinflammation use these cells and use specific compounds for positive controls of M1 activation. And so they'll use LPS or lipopolysaccharide as a positive control, because when your immune system is exposed to lipopolysaccharide, which is a bacterial cell membrane component, then it will cause an immune response. Or polymorbyl 13 acetate or interferon gamma. These are all things that cause neuroinflammation in the brain via response from the microglial cells. However, researchers across the globe has also run into issues with HMC3 cells specifically failing to become activated when they use the same conditions that they use for other microglial or macrophage type cells that are described in literature. So if you go on to like ResearchGate or any other forum where researchers can discuss their work and try to troubleshoot as a community, many of them will say, hey, I am using these HMC3 cells. I am dosing at 10 nanograms per milliliter of LPS and I'm not getting activation. And someone will say, well, try PMA, that works for me for my microglial cells. Someone else will say, I tried that, it's not working, but I use this instead. And so there's just not a consistency, which can further lead to the reproducibility crisis that is in science right now. And so the goal of our study was to find a prototypical stimuli that produces a consistent M1 phenotype in these cells. And I hypothesized that possibly it could be in a 4-hour exposure. And I thought of that because the microglial cells are very metabolically active and they proliferate very rapidly. So I'm assuming that there might be a chance that microglial cells are becoming activated when we put LPS or PMA or interferon gamma into their culture to activate them. But before that 24-hour time period that most people use, they're clearing up and reverting back to the M0 or surveillance phase. So the methods I used was I dosed these cells as said at 4-hour time points with the following reagents. These were the ones I figured would be best for it. I used 10 micrograms per milliliter of LPS, 10 micrograms per milliliter of LPS combined with 100 nanomolars of PMA, and as well 10 nanograms per milliliter of interferon gamma and 10 nanograms per milliliter of interleukin-1 beta or IL-1 beta. After that, I harvested them and I stained them for both IL-6 or interleukin-6, which is a cytokine released from the cell, as well as INOS or inducible nitric oxide synthase, and then ran them on a FACSverse, which is a flow cytometer made by BD, and analyzed them using FlowJo. So it's a very high-powered system, and about every sample we get will give us anywhere from 100,000 to 200,000 events or data points. And so it's a very high throughput, high data system. And so because of that, you also have to sort the data you get. So this is what happens is whenever we get our data, it comes out like this format right here. So we come out and we first look at the scatter of the cells when we arrange them, and we have to make sure that no debris came through. Because even though if we have a sample, like a 1 milliliter sample of cells, there's going to be debris from cells falling apart, antibodies that didn't bind, or reagents just floating. So I gated for the size here. And after that, I sent them to viability because, well, I'm interested in looking at the cells. I want to make sure I'm only looking at live cells or viable cells. So I use something called fixable viability stain 780, which is an amine-based dye that enters into the cell when there is a dead cell or a permeable membrane to the stain, and stains the open amines on the proteins in the cell. So if it stains the open amines on the proteins, that means the cell is dead and not viable. Therefore, I took everything below a certain threshold. And after that, I looked at their IL-6 fluorescence and gated it, saying that at 10 to the third fluorescence values and greater is a positive expression of IL-6, while below that is a negative expression. And as you can see here, I also talk about it. And after I gated my cells, I looked at the results for it. And as you can see, there is an increase in expression of interleukin-6 in all of the stimulated groups after 4 hours of exposure. But among the stimulated groups, there's no difference in IL-6 expression. So as you can see, both LPS, LPS plus PMA, and interferon gamma plus IL-1 beta all have the exact same rate of activation compared to the unexposed. And while those do look very similar, this is on a logarithmic scale. And so it is already an entire log unit greater than the unexposed control. When I looked at INOS or inducible nitric oxide synthase, there was very just wide results, and there's no noticeable difference of expression of INOS after 4 hours of exposure. And while you could say that because it's above that same threshold, it is a positive expression of INOS, once again, these HMC3 cells have, well, they've been characterized for being primary microglial cells that have been immortalized. It might be that there have been some differences in their genetic expression and their reaction to certain stimuli. And so there's also the issue of earlier research suggests that INOS is a strictly pro-inflammatory macrophage product. So when it enters into the M1 phase, it begins production of INOS or the usage of INOS. But now more current research also shows that it is very commonly an anti-inflammatory product. So more research needs to be done on figuring out whether or not INOS in HMC3 cells is a pro-inflammatory or anti-inflammatory pathway. After that, we also looked at 24-hour time points because that's what most people do. And as you can see, once again, there is no difference in activation. So this further proves the point that these cells at 4 hours, as you saw earlier, showed positive activation, but at 24 hours, it was reverting back to the M0 phenotype. So once again, this shows that these cells, probably due to their metabolic activity and rapid proliferation rate, are reverting. So our conclusion is that 10 micrograms per milliliter of LPS, both with and without 100 nanomolar of PMA, as well as 10 nanograms per milliliter of interferon gamma plus interleukin-1 beta are appropriate M1 activators for HMC3 cells, but only during a 4-hour exposure. If you would like to do a 24-hour exposure for these controls, a different compound needs to be assessed, perhaps something that will either stay in the HMC3 cells for longer and not be metabolized as quickly, or possibly a stronger dose that does not approach the LC50, or just another compound overall that might cause a stronger reaction. And so for the future, what I'd hope to do is assess the functional cytokine expression using proteomic multiplex immunoassays. We do that with our Magpix instrument that we have. We can take a look at around 48 different cytokines and chemokines based on just 100 microliters of fluid. We also want to look at finding the optimal expression, the optimal time frame for the expression of activation. So I only looked at the 4-hour time period. It would be helpful to look at the 6, 8, 10, 12, all the way up to 24-hour time point to find out when the highest level of activation is. to find out what is the most optimal time to dose these cells to get a positive control. And also it would be useful to assess the M2 macrophage phenotype under different compound exposures as well to find a standard for M2 activation for HNC3 cells, because these are wonderful cells for the study of neuroinflammation, and it would be great, further research needs to be done into them to make sure that they are proper models. And so I'd like to thank Dr. Bruce for her support and guidance as my PI. I'd also like to thank Grace Aquino, our doctoral candidate, for her mentorship. And I would also like to thank Rishi, Manu, Caitlin, Katie, Nazil, Morgan, and Jeff, and all the undergraduates of the Bruce Lab for their support in completing this project. I have my references here if anyone wants to take a look at them. But other than that, are there any questions I can answer?
Great job, Bennett. Let me, am I muted? No, sorry, y'all. Can I ask a question?
Sure.
One question that maybe you can clarify for the judge is how did you select the stimuli that we did, and how did you select the target or the markers of activation intracellularly that we did use?
Of course. Okay. So the first thing that I did was actually, Grace, could you mute yourself? Thank you. So the first thing I did was I went through a literature search and looked at everything I could on microglial activation, both in HMC3 cells, in other immortalized or primary microglial cells, and made a list of all the compounds that people were using as positive controls. After that, we did some beginning experiments using the 24-hour time period, just to see if anything showed any sort of difference. We tried things such as 0% fetal bovine serum, so not adding any serum to the media to cause activation. We tried at least 4 or 5 different concentrations of LPS, including very high concentrations. We also tried other various compounds as well that did not turn out, and found that these 3 were the best and most optimal ones, and we continued our research into those. And to looking at the cytokines and chemokines themselves, we initially were looking at surface markers, however realized that in our process for harvesting and preparing our cells, we're using an enzymatic detachment agent to bring the cells off of the plastic flasks, and those enzymatic detachment agents also cleave not only the proteins that adhere the cells to the flask, but also all the proteins on the outside of the cell. So those surface markers we were looking at were being cleaved off, so we're getting no instance of activation there because there were none there. So we had to switch to an intracellular marker, which we found would be a good one would be IL-6 because it's commonly studied as well in neuroinflammation.
Thank you. Good job. I have a follow-up question, but I'll let the judge ask if she has any, and if we have time.
Yeah, I do have one question, and that just is why you settled on the 4-hour time point as your initial time point to look at. Like why specifically 4 hours? Why not somewhere in between, like 12 hours? If you're, yeah, that's my question, sorry.
That's a good question. So what I was curious about was, like I said, I knew they were very metabolically active, and I wanted to see if there was a big difference, because I know that obviously with the immune response, there's going to be something that the moment that foreign compound touches the cell, it's a signal. There's a cascade of signals through the cell. And so I want to make sure I had something that would be able to be read pretty quickly. And so since the immune response is very fast for the compounds we're looking at, or the markers we're looking at, I want to make sure that they would not disappear. And to be honest, I thought the 12 hours might have been a little long. And so we just shortened it. And so caused a lot of long days, but we just decided that 4 hours would probably be optimal, but we would love to go back and take a look at other time points as well to see if maybe 6 hours is best. It might be that the activation response is just ramping up at 4 hours. And so further work would need to be done in that area.
Great, thank you.
Is there time for one more question or no?
I think that we're going to be going over time.
Okay, then I'll hold my question.
Okay, so now thank you, Bennett. And next we have Caitlin presenting her topic. If the judges are ready, and if you are ready, please share your screen.
Awesome.
Hi, everyone. 1 second. Okay, so I'm going to be going a little fast just for time's sake. But my name is Caitlin, and I also get the opportunity to work at Bruce Lab in the Environmental Science Department at Baylor. And my project was evaluating the effects of acute diesel exhaust particle exposure on P glycoprotein function and expression in the blood-brain barrier. So first up, I want to talk a little bit about some background, particularly what is diesel exhaust particles and how does that affect us in our daily lives. So diesel exhaust particle is, as mentioned before by Maziel, a xenobiotic, so basically a foreign material that is not natural in any way. It's mainly made of metallics, and it is a product of diesel combustion. So here I have like a little diagram that shows that air pollution is actually the major portion of air pollution that is hazardous to humans is diesel exhaust particles. And it very interestingly has two routes of entrance into the human body. So first there is the direct route, which is basically, as Maziel mentioned, through the olfactory bulb and straight into the blood-brain barrier at like the skull. So basically straight into the brain, or the indirect pathway, which is shown here as going down into the lungs. And if you look, I have a little diagram that shows microglial cells taking up the DEP, and basically when it realizes what this is, it will make what we believe pro-inflammatory cytokines that can then decrease phagocytosis and can increase cell activation by other cells. So in these diagrams, you can kind of see what I've made is like DEP has entered the bloodstream, and then it will exit through these cells, or it will try to go through the cells and get into the blood-brain barrier. However, P glycoprotein, a very important protein in the blood-brain barrier endothelial cells right here, actually will pump out any foreign materials that are trying to enter into the brain back into the blood. So this pump is very important for a lot of chemotherapies as well as many drugs, because it is a multi-drug resistant protein. So basically it has the ability to bind with multiple different foreign bodies, and it can pump them back into the blood. And this has been a major area of research because a lot of chemotherapies, which are very important if you're trying to diagnose or work with the brain or spinal cord, it can be very important in the chemotherapy administration as well as absorption into the actual area that needs to be treated. So we wanted to ask for this research project, how does acute DEP exposure affect both the P glycoprotein in a monolayer of endothelial cells, as well as in a co-culture of both human endothelial cells and microglial cells? Because as mentioned in previous presentations, microglial cells are the immune cells of the blood-brain barrier, and they can actually act as a pro-inflammatory cell and can influence the blood-brain barrier and its function. So first up, I just kind of wanted to mention our cell culture. So basically for D3 monoculture, D3s are our endothelial cells, and they were a major, very important immortalized human endothelial cells that help us view basically in vitro form of the blood-brain barrier, because we can't exactly ask people to inhale diesel exhaust particle for research. So for now, we're using human endothelial cells in cell culture. And then you'll notice on the second plate that there are much bigger, like darker cells. They're not necessarily darker, but they absorb the light slightly differently. And those are our HMC3 microglial cells, which actually, when in co-culture with D3s, form a much more rounded cell and are very much spread across and sit on top of the endothelial cells. So first up, we actually did immunocytochemistry. and we used antibodies that fluoresce to demonstrate specific production of P-glycoprotein. So in this first row, you can see there's like a blue, like very blue, and this is actually DAPI, which is just a blue fluorescence for the like DNA basically of a cell. So this is the nucleus. And in this second one, you can see it is DEP in 1% DMSO. And if you look in this middle row, you'll see there's very little green in the middle or in the top and bottom rows, but there is more green in the center. This is actually exposing P-glycoprotein. So the green is P-glycoprotein. So if you look, you'll see DEP is actually brighter, or P-glycoprotein is brighter when exposed to DEP, which means that their cells are producing more. So they're producing more protein when exposed to DEP. However, if we look at what's called the rhodamine 123 assay, we will find out something more interesting. So this assay basically was you basically use a fluorescent material and it's absorbed into the cells, and then the P-glycoprotein is supposed to pump the substrate back out of the cell because it doesn't want that material to get into the brain or the spinal cord. And the reason that this is interesting is because tariquidar is a known inhibitor of P-glycoprotein. And basically this graph is showing fluorescence. So this fluorescence is going to increase with the amount that's staying inside the cell. So basically if P-glycoprotein is not working, more fluorescence will be seen inside the cell. And so this is showing the functionality and the of the protein. So if you look, you'll see the control cells are pumping this rhodamine out at a normal rate. However, when exposed to DEP, the P-glycoprotein looks much more similar to the inhibited cells. So that means that it is basically not, when exposed to DEP, the P-glycoprotein is not pumping out any of the rhodamine 123. Which brings us to our last test that we did, which was RT-qPCR. And basically, for this, we had to take cells that were either exposed to DEP or not exposed to DEP, and we would extract their RNA, convert this into cDNA, and then use the cDNA to determine if any, if the protein is being transcribed more frequently, basically. And interestingly, we found that there's very little difference in a monoculture of endothelial cells, and in a co-culture when exposed to DEP, they actually decreased the transcription of mRNA for P-glycoprotein. Now, this was very interesting to us because it kind of indicates that with all of these materials together, we find that P-glycoprotein is being more frequently produced by the cells. However, they're not functioning. So a theoretical process for this might be that the cells are trying to remove the cytotoxic DEP. However, they are unable to do so because of P-glycoprotein inhibition. And so they're trying to produce more P-glycoprotein. However, the proteins become non-functional as soon as they get to the membrane. So they are not actively working to do anything. And microglial cells, when in combination in this co-culture, could be acting in some form to inform like the endothelial cells that it's not helping and that they need to either produce some other type of protein or they need to find some other way to work. And so they might be in a unknown route or mechanism informing the endothelial cells to lower their production of P-glycoprotein. And the reason this is so significant is because it can really affect the human body in their ability to expel foreign materials. So if this P-glycoprotein in the blood-brain barrier is non-functional, there can be a significant accumulation of toxic material in the brain and in the spinal cord. And this has been associated, and this dysfunction of the blood-brain barrier has been associated with neurodegenerative degenerative disorders, and can actually be very important in the use of chemotherapies, because even though chemotherapy in this condition would be able to stay in the brain longer if DEP was causing this, it would never leave the brain or the spinal cord, which could have significant injury as well, because it is assumed that most toxins and chemotherapies would eventually be degraded by the body. However, if that's unable to do so because it's unable to leave the brain or the spinal cord, this can also cause significant damage to those areas. And last, I would like to say thank you to everyone, including our fearless leader, Dr. Erica Bruce, as well as my amazing graduate student and doctoral candidate, Grace Quino, and all undergraduate students of Bruce Lab. These are my references. These are just a few of my references. There are many more, but these are the biggest ones for this PowerPoint. And that brings me to questions.
Great job, Caitlin.
Thank
Congrats.
you.
I have, of course, a question. Maybe could you take a minute to explain for the judge how you did the rhodamine assay, just, general?
Of course, yeah.
And then, sorry, the second part of that is why it's a specific, why the rhodamine assay is a specific measure of P-glycoprotein function.
Of course.
Yeah.
So for the P-glycoprotein, we did a rhodamine 123 assay. And basically, this is where you take a fluorescent, which is rhodamine 123. And initially, we had to troubleshoot this a little bit, but we found that adding rhodamine 123 to the cells for, I believe we did 6 hours. And we let the cells absorb the rhodamine 123, basically how they normally would. And then we added an inhibitor or DEP, or allowed the cells to continue, and saw if the cells were able to expel the DEP or the rhodamine 123, I'm sorry, to see if they were able to expel the rhodamine 123, because P-glycoprotein is mainly an efflux protein, so it will mostly pump things out of the cell. It doesn't really take things in very often. So we did this in order to show that P-glycoprotein is functionally pushing things, like removing things from the inside of the cells. And if exposed to DEP, this was not happening, that meant that there would be an accumulation of fluorescence inside the cells. So then we removed the excess liquid and we lysed the cells, and we used a fluorescent image screen. I can't remember what exactly it's called, I'm sorry. But we used a fluorescent.
A plate reader.
What?
A plate reader.
A plate reader, basically, yes. And we just looked to see how the fluorescence changed based on the different groups, whether or not they're exposed to DEP, or if they were control group or exposed to a known inhibitor.
Awesome, thank you.
Of course.
Do the judges have any more questions? Judge. Okay, so if not, then we thank you, Caitlin, and we can move on to the next and last presentation. So Madison, if you're ready and if the judges are ready, you can share your screen.
Thank you. I apologize. My PI had me under the assumption to prepare a poster. Can y'all see it?
Yes.
Okay, awesome. So hello, my name is Madison Brajal. I'm a sophomore at Baylor University. I work in the Lovato lab in the environmental science department, where I started research this past fall. Oops. Okay. Today I will be discussing and sharing the results of the in vitro, totally got out. Of the in vitro toxicity of 5 indole compounds on 3 human cell lines, one being HepRG, which are liver cells, MSC, which are adipose-derived mesenchymal stem cells, and MRC5, which are lung fibroblast cells. So we commonly hear people talk about our gut health and what's good for our microbiome. So with an increased research interest in the past 2 decades, more information on the relationship between diet, gut microbiota, and their host has been attained. New data suggests that the amino acid tryptophan can be metabolized by our microbiota and lead to the synthesis of several indole compounds. So in this experiment, I tested 5 indole compounds, indole 3 carboxylic acid, which is I3CA, indole 3 aldehyde, which is I3A, indole 3 acetic acid, which is IAA, indole 3 propionic acid, which is IPA, and 3-methyl indole 3-MI. So after exposing the different cell lines to concentrations ranging from 1 picomolar through 10 micromolar for 24 hours, I added the MTT assay at the 20-hour mark and read the plates at the 24-hour mark. The graphs represent the lethal concentrations of the 5 compounds. I3CA, IPA, and 3-MI showed strong cytotoxic effects in stem cells with LC50s ranging from 0.2 through 1 micromolar, while there was very little toxic effects shown in HepRG. I3A also exhibited high cytotoxic effects in both HepRG and MSC, while IAA only showed significant differences in HepRG with LC50s of 2.21 plus or minus 0.32 micromolar. IPA showed LC50s of 0.4 plus or minus 0.14 micromolar in MSC and 0.91 plus or minus 0.1 micromolar in MRC5. But interestingly, IPA presented no toxic effects in HepRG with LC50s above 100 micromolar. Overall, stem cells appeared to be the most sensitive to the indole derivatives compared to liver and lung fibroblast cells. Indole derivatives found in human plasma tend to be in the 1 micromolar range. Due to some LC50s being in that range, risk levels should be further studied and evaluated in human toxicology, we believe. This study showed that several indole compounds may exert toxic effects in the selected cell lines, with these being indole and cell type dependent. Indole concentration is overall dependent on the amount of tryptophan in the bacteria species, which is more abundant in the microbiota. Follow-up studies should be conducted using additional cell lines like brain cells or intestine cells, which would help establish a better understanding and characterize their influence on human disease etiology. This would also be benefited by examining the mechanism of action of the indoles. And then lastly, I would just like to thank my principal investigator, Dr. Ramon Lovato, our graduate students, Megan Salon and Ashley Ball, and all of the other undergrads in the Lovato lab for all their help.
Okay.
Great job, Madison. Oh,
sorry.
sorry. Can I ask a question? Good job, Madison. Thanks for that presentation. I have one question. I obviously am not an indole person, but okay, one question. So what are the health, because in your title it says that indoles are associated with, potentially associated with disease, human disease. So what are the health risks or the adverse health effects that are of concern from indole toxicity? And then the second side to that question is, are there any beneficial roles of indole in the gut? Because, well, my understanding is, the microbiome produces all these metabolites or breaks down tryptophan and other amino acids to allow the absorption of different nutrients. So is there a beneficial role, like a need for indole?
Yeah, that's one thing that we don't know. And so that's why there's very little research on indole specifically recently, because obviously, like I said, like in the past 2 decades, there's been a growing interest in learning about the gut microbiome and what's going on and what's beneficial and what's not. And so looking at indoles, it's just one of those things that's less looked at. And so we're not sure if there are any health benefits to it. For this study specifically, we just looked at the cytotoxicity. And so from there, we were able to see which compounds did show toxic effects. But I feel like it would need to be further studied to find if any served any beneficial roles in the biome.
Okay, thank you.
So if the judges don't have any questions, that concludes our presentations and concludes the environmental science toxicology division. So we're going to take a picture of everyone involved for social media. So if you don't want to be in the picture, just turn your camera off. Okay. 3, 2, 1. Okay. And then, so we only really have one judge, so you don't have to stay on to deliberate, but do turn in your Google Forms or however they're having you do your judging. And so awards will be announced after the keynote speech today, and feedback will be emailed after the conference is over. The next event is the graduate fair, which should be starting in a couple minutes. So you guys can refer to the program to find the link for that. And yeah, thank you everyone for attending and presenting and judging.
Great job, y'all. Thank you.
Thank you.
Yeah, nice job, everyone.